this. There we go. Okay, and let me open up. Hold on, sorry. I have a new monitor and it's kind of blocking everything. Okay. I'm going to share my screen. Let's see if this is gonna work. Oh. Okay, do you see that?
Yes.
Okay, great. So I think while I'm sharing, if you have questions, you can speak up or message in the chat. I'll look at the chat as well while I'm presenting. So I have 22 slides. At the very end, I'm going to actually switch over to Firefox and just show you how I look at papers. I think this will be about 40 minutes, and then we can just do a bunch of questions, specific questions if you want. This is from Sound Bio Lab, so that's why there's a logo there. But yeah, it'll be interesting. I'll cover everything from why do we do literature review to what are the parts of a paper to what do we do on the internet to find papers. Okay. I might skip through some things really fast. Again, I'm gonna share with you guys a slide so you can look. I would really encourage you guys to look at all the slides later, because I think this is a useful skill. Like it's a practical skill to have. So yeah, okay, literature review. Literature review, what is it? So first of all, it's definitely to identify existing research and gaps in a topic. I think most people don't think about the gaps. It's obviously to read what happened before, but it's also really useful when you find out what hasn't been done, right? So if we're trying to do something new, we obviously need to know what hasn't been done. And that's ultimately the answer we're trying to get to. By learning what exists, we're finding out what the gaps are, okay? So real deep literature review is actually trying to figure out this. What is not done yet? Literature review allows us to formulate a research question and planning a project based on what's happened before, right? So that kind of gets added into your understanding of why is a certain project necessary to do. And learning. So by doing literature review, you can see literally what experiments did they do, what instruments, what equipment do you use, what works and what doesn't, so on and so forth. When you're doing iGEM project development, you actually don't really do traditional literature review. So if you have to do literature review in a class or something, you will do it in a really different way than what iGEM looks at and what I would recommend for iGEM. And the reason why is in a regular literature review, you will actually read the paper. In iGEM, you probably don't want to read the paper most of the time. Instead, you're actually trying to find specific answers to specific questions and information that either supports or doesn't support or kind of disproves what your idea for your project is currently. Okay, so overall, that's the goal of literature review. You're trying to figure out if your project either makes sense or if your project needs to be tweaked somehow. Literature review is supposed to help you do that. Literature review in the simple terms is how to read a scientific article, right? And an article is many different things, which I'll cover later. Well, how do you read a scientific article? It actually is based on your objective. So anytime you look at a paper, you actually need to have a very, very clear objective in mind, okay? What do I mean by that? Well, I will probably get to that later in the presentation when I actually show you what I'm doing, and I'll walk through what am I thinking. But first of all, let's talk about the anatomy of a research paper. So what is a research paper and what is it comprised of? So experimental research papers, which are the majority of scientific literature out there, share original work. So anytime there's a research article, So most experimental research papers are just called research articles. They're usually always original. So you're technically not allowed to publish on something that has already been done before. That's plagiarizing, right? So it's always new work or new additional work to disprove or support something. It's often extremely very specific. So it's on a certain subtopic with a certain use case in a certain way or something like that. So if it's, I don't know, synthetic biology, maybe it's talking about the synthetic biology of genetically engineering this species of frog and this specific gene alteration in this frog for the sake of, I don't know, muscle enhancement. I'm just making this up, but something specific like that. It will reflect the scientific method that the researchers used, and it has a standard format almost all the time. There's usually a little bit of tweaks to the format, but there is a overall standard. Okay, what is the overall standard and how does that fit with a scientific method? That's what I'm going to talk about next. So this is the overall scientific method that you probably learned in like high school. A paper is structured very similar to a scientific method, but in an overlapping way. So these are the sections of a regular research article. Okay, introduction, materials, methods, results, discussion, conclusions. There's usually an abstract and there's also the title, but that's out of what we consider the body of the text. So these stuff that's in this table are what's considered the body. And they relate to the scientific method in many different ways. So in the introduction, there's usually comments about what is the observations that people made, that the researchers made, what is the background research that was made, and most importantly, what is the hypothesis? What is the prediction being made? Okay. In the materials and methods and the results, they will usually talk about what did they actually do? What did they actually test? In the discussion section, they will talk about their analysis and what are their conclusions that they're drawing. Note that it's not the conclusion section where they're talking about the conclusions of the experiment. They will report the results, whether or not the hypothesis was supported, usually in the discussion, sometimes in the conclusion. The conclusion is overall basically like a summary of what they did in total, with an emphasis on what are the conclusion in the sense of like conclusion of the story, not conclusion of the experiment. What are you supposed to get out of this whole experiment? The conclusion is also often, sometimes a discussion, where they will actually talk about what are the new observations and questions that they have now. So for example, based on this experiment that was done, do they have any new questions, right? What can be done in the future? Okay. So zooming out a little bit from the body, so there's also the abstract and the intro. There's also the abstract, right? This doesn't say title, but the abstract is kind of the addition here. The abstract is really, really important. It's basically a summary of what is the most important parts of the paper. It's not a summary of the paper. It's a summary of the most important parts. So they actually do a lot of work for you to make the abstract. The abstract is usually written last after everything else in the paper is written. And the abstract is supposed to be like a Cliff Notes version of the paper. So you can probably imagine how useful that would be. Okay. I already discussed this, but this is another way of thinking about these sections, okay, in a very, very simple way. After the body, there's usually the acknowledgement sections. The conclusion is considered sometimes out of the body. Acknowledgement sections, literature cited, and supporting materials. These are also very important. They are not part of the body, but they're very important. Acknowledgement is basically who gets acknowledged for the work in the paper. So all the researchers that does experiments, the person who writes the paper, who edits the paper, maybe they had to ask another lab next door for a piece of equipment. That's all written there. They have to cite every single person that has helped them out in the acknowledgements. Literature cited is what papers are they referencing in their own paper. So like I said, in the introduction section, they'll talk about what has been done before, right? When they reference what has been done before, they have to put citations in every single claim. So that's the literature cited. It's a list of all the literature that they cited in any part of their paper. Supporting materials are sort of like appendices. So sometimes they will have a huge Excel file, or they'll have like additional pictures, or they have a video that they want to attach that's part of the experiment. That's what the supporting materials has as a list. Usually the supporting materials is literally another file that you can access through a URL or something like that, or maybe like a YouTube link even. So that's supporting materials. These are all important because these will be very helpful for iGEM. Okay, so you need to develop a reading strategy. A reading strategy is unique for each person, and reading scientific articles can be pretty complicated. What you don't want to do is read word for word from start to finish. That is not efficient at all. Most people actually skim it, and they don't skim everything. They actually skim specific sections of the article. So they usually go, actually, I'm just going to click next. There's a table here that's actually really helpful. In general, people will skip right from the title to the visuals. So like the figures and the pictures and the graphs. Why? Because usually they're experts. They don't need to know the introduction. They don't need to know why it was done. They're usually experts in the field. They already know that. What they're interested in is, what did this experiment do? So they go to the results, which are usually in figures and pictures and stuff, right? We're not experts, so I would recommend that you start with the title and then the abstract. Why? Because the abstract is a really good summary of the paper, right? What did they do? If you read the abstract and you realize it's not what you're looking for, you can move along. Just stop reading the paper, find another paper, right? If it is something that you're interested in, then you actually go into the whole process of thinking about the reading strategy. What should you go to next? And I will talk about that as well. Okay, I want to explain a few things about the sections, specific sections, okay? The results section. The results section is not an interpretation. It's not a comparison. It's just the facts. So in the results section, they will not talk about this was interesting, or this is significant, or this is what we were trying to look for, or this proves a hypothesis. It doesn't say any of that. It literally is a listing almost of what happened. What results did they get? Okay. The discussion is where they talk about, why is this significant? Why was this important? Did this prove or disprove what we were looking for? What other questions do we have? They say stuff like that in the discussion. The discussion usually starts small and goes big. So it begins with an interpretation of the results, the data, and then it will actually start getting broader and broader, in the sense that they might start making more bigger claims about what the data suggests or might suggest. They might start comparing their data to other literature, that other experiments that were done in the past. And then they might even start introducing things that sometimes is in the conclusion, sometimes it's in the discussion, about future steps, such as what should be done next, what other questions have come up, how does this impact the field, stuff like this. An introduction is the opposite, and I'm deliberately not going in order here. An introduction is the opposite. An introduction is the discussion, but flipped. It starts really broad and then narrows down to what is their thesis statement, basically. What is their hypothesis? What is their prediction? So they will start, as synthetic biology is a field that does blah, blah, blah. And then it narrows down to prior literature has looked into various frog species and found that these genes are blah, blah, blah. There's a specific gene that controls muscle movement, and prior studies in this gene has been unclear on how this gene really affects muscle growth in frogs, or something like that. So it specifies down and down towards their, at the end, their specific experiment that they're doing and why do they have to do that. The abstract, sort of like I said before, you can think of it like the advertisement for the article as well. There's two questions that mainly gets answered in the abstract, in a good abstract. One is what has been done and what are the main findings. And so when you're looking at an abstract for a paper, it's good to ask these two questions and even like highlight for yourself what are the sections of the abstract that answer these two questions. Usually abstracts are kept as brief as possible, usually less than 250 words, not all the time, but they're usually pretty short and they're scrunched up in one big paragraph. So, okay, actually, I'm gonna pause for a second. Do you guys have any questions so far? Give me like a thumbs up or something if you don't.
I don't have any so far.
Okay. Anyone else? Okay, cool. Great, I'm gonna keep on going then. So this is an abstract that I found. It's a pretty good abstract. So you might look at this and be like, whoa, that's a lot, okay? So we can try to digest this a little bit. First of all, let's look at the title, sorry. Relationship between the lability of sediment-bound metals and their bioaccumulation in benthic invertebrates. So I don't know about you, but there's some terms in here that I don't know. I don't know what lability means. I don't really know what benthic invertebrates are. I kind of get the sediment-bound metals. They're talking about cadmium, copper, and zinc. Okay, I get that. Bioaccumulation means that these metals are kind of gathering in the invertebrates, I guess. So they're invertebrate animals, and I guess these metals go into these invertebrates, right? So there's a relationship between this lability thing of the metals and how much these metals go into these invertebrates. So it's probably talking about pollution, right? About maybe the contamination of these and the accumulation of these metals in animals. Okay, so that's my guess, looking at just abstract. I mean, title. Okay, so now I'm gonna look at the abstract. And I'm gonna take the abstract and kind of highlight what is the section that talks about what has been done and what is the section about what is the main findings. Okay, so I've done that here. So you can see that almost all the abstract is what has been done and what are the main findings, right? Basically, every single sentence, you can highlight something in these two categories. So that shows you that this is a good abstract because it focuses on the two questions that I brought up, right? So it investigated metal contamination. So let's digest this. The researchers investigated metal contamination in 9 sites. Okay, so it is about contamination. Okay, 9 places. Both total and labile concentrations of metals are determined in superficial sediments. So I still don't know what that is, but maybe that's a specific subset of like a type of metal concentration, right? That's what I'm guessing without Googling. In actuality, I would probably Google that immediately. The influence of different pHs was tested, and metal lability at pHs encountered in the gut was compared. So that's what they're actually testing. That's what they're actually looking at in the experiment, right? So that's what has been done in this experiment. They looked at 9 sites. They looked at these 2 concentrations of the metal, and they looked at that concentration in the sediment, right? And then they tested the pHs in the gut of the animals, the invertebrates, right? And I find out, oh, okay, here, there's this thing called a ragworm, the blue mussel, and oysters. So these 2 are clams, I know. So that kind of invertebrates, okay? So I'm trying to figure out what is happening in this paper. What did they find out? Well, here they're talking about results. Cadmium showed highest liability and copper the lowest, whereas zinc liability was intermediate. I know this is a results because it doesn't say anything about this is amazing or this is significant or this is bad. It just says this is what happened. More of that here. They also say this relationship did not always hold for copper and zinc. So they're talking about what they mentioned in the title, right? And then they move on to the actual discussion. Procedure proposed here to assess metal bioavailability seems a significant improvement of the existing methodologies of risk assessment. So what do they mean by that? The way that they tested metal bioavailability here is better than what used to exist. Great, that's a conclusion that they made, right? So that in about 5 minutes, I skimmed the abstract and I found out what they did and what did they find out, okay? So that's what you should do in the abstract. Any questions so far? Okay. I feel like I'm just talking to myself, so let me know if you have any questions. Okay, now I want to talk about the different types of scientific literature. So most papers are experimental research papers, but there's also things called reviews. Sometimes they're called special features or highlights, and these are really nice as well. There is sometimes books and chapters that you can find. There's sometimes protocols, letters, and patents, okay? There's also something called preprints. I should write that here actually. And I'm gonna cover each type. So from experimental research papers to books and chapters, they are all describing experiments, but it goes from specific to broad range. So what I mean by that is an experimental research paper is always talking about one research that was done by a lab, right? Or maybe a collection of labs, but it's one research. And it's always new. So it's usually about a new discovery, a new method, or a new tool. It's very, very specific. A review or a special feature or a highlight is actually a collection of experiments. So these kinds of papers are actually really, really useful if you want to get a broad idea of what's happening, right? So reviews usually have a very specific scope, like, I don't know, gene modification in frogs. Let's go with that example, right? But they will, if you find a review on that, they will actually like cover all the different kinds of experiments that have been done in the past 10 years for gene modification for frogs and talk about what are the trends that people are showing or what has been working in multiple different experimental research, right? What has been shown to not work, or what is the meta-analysis of all the kind of conclusions that were made. That's the kind of things that a review shows. So it's actually very useful for us, for iGEM. A book or a chapter is much broader. It's almost like a textbook, right? And it often is like a textbook. It often doesn't specifically talk about, it doesn't talk about specific research. It rather talks about things in general, about the field. And then there are papers that are protocols. So a protocol is just literally a protocol that somebody made. It's a way of running some sort of experiment or something like that. So it's usually attached to a research, but sometimes it's published separately. A letter is sort of like a preprint. Letters are rushed out when like people are excited for it or when the researchers feel like it's really significant and like the paper isn't finished yet, but they want to let the world know like what's been happening. That's a letter. They're usually extremely short, like 1 or 2 pages, and they're written like an editorial piece like you see in a newspaper. Sometimes they also use letters as responding to papers, like literally like there are letters written to say, I totally disagree with what you did and your research was dumb. Like, or your research was completely flawed. I suggest you retract your paper, something like that. There's things like that that happen as well. Patents are patents, they're inventions. They are scientific literature though, because when you make a patent, you actually need to describe everything, well, basically everything about what you're patenting, right? Enough such that people can understand that what you made to make sure they don't make what you made, that you can hold intellectual property. Oftentimes patents cite papers and papers cite patents as well, especially for engineering. Okay, what you're trying to do for iGEM is not searching for papers, it's really reviewing papers. So how you review changes how you search, okay? What I mean by that is we're going back to the objectives thing. based on your objective, how you might look for something changes, and how you might even Google searches. So first of all, you need to define your objective. What is your objective? Is your objective simply for finding out more about this field? Are you open to anything in this field? Are you trying to find more professors, more experts in the field? Are you trying to find a protocol that you can't figure out? You know, all these things change how you're looking for it, right? So for example, if you're broadly looking at a field and you're not so sure, instead of looking at papers, you might start at Wikipedia, right? You can totally do that. Let's say you're done with Wikipedia or it's too specific. Then you might want to look for a review, right? Or a special feature or a highlight, right? If your objective is rather, I want to find out how prior research has done a particular experiment, then you're looking for protocols, right? Or you're looking for specific experimental research papers that did specifically what you're also trying to do, so you can look in their methods section and see how they did it, right? Because they will explain that in the experimental research papers. Other than that, the table is basically to keep in mind certain things, tips. Okay, so now we're gonna look at the paper. I'm making really good time here. So before I move on to looking at a paper, do you guys have any questions over what I covered?
I had a question.
Yeah.
So our reviews, are they, when they go into the results, do they each do they separate the results by the papers or by the results? Do they like pair them up or group them up in regards to the type of results that they're getting?
Yeah, that's a good point. I forgot to say that the structure of a regular research paper that I covered only really applies to experimental research papers.
Oh, okay.
So for anything else, they may follow the structure of a research paper, like the introduction, methods, results, discussion, conclusion, like that.
Okay.
But they may not. Reviews and special features often do, but they also don't. When a review follows that format, it's usually because they're doing a meta study. So sometimes a researcher or like a really big expert in the field might do a meta study where all they're doing is they're collecting all the experiments and doing sort of a experiment to see what can be figured out from all these collections of research, right? So they actually take all the data from various different research that was done and run like a meta-analysis, like a data analysis on it to see like maybe they're collecting 3 different data sets from 3 different research projects that were done, right? And trying to come up with like one big total or something like that. When they do stuff like that, they usually have a very clear conclusion section about that meta study. Does that make sense?
Yeah, that makes sense. That makes a little bit more sense.
If they're not doing that, if they're doing sort of like an overview of various different research that was done in a much more qualitative way, where they're trying to basically be like a nice person to the reader and be like, this is an overview of this field in case you don't know it. If they do something like that, they probably won't have like a conclusion section. Okay. Instead it will feel more like a book, more like a textbook.
Mm-hmm.
Yeah. Any other questions? Okay, cool. So I'm going to move into looking at a paper. So while I'm doing this, ask me a whole bunch of questions if you don't understand why I thought a certain thing or I did something, okay? I'm gonna try my best to explain all my thoughts as I'm doing things to sort of give you a simulation of what to go through and how doing literature review is like, okay? I'm just gonna preface and say that this literature review process is very active. It is not simply click a link, read it, click a link, read it. It's constantly thinking about what you're looking at. And so some people actually think it's pretty fun. it's cool to look at all these kind of research and find like the right one. It's almost like a puzzle where you're trying to find like the right paper. So I want to try to show you how active of an activity this is. Okay. So I have an example. Wow, that pink is really stark, but whatever. I wanted to find a paper on using microfluidics and synthetic biology, see what has been tried, and if there are any professors I can contact. I'm writing this out. This is my thought, but this is a complete thought. Why? Because I know what I want, microfluidics and synthetic biology, and I know what I want to try to find out, which is what has been done. Are there any other professors? Okay. So I'm not looking for protocol. I'm not looking for something specific. I'm still at a vague kind of topic. And I'm just trying to figure out what has been done before right now and if there are any professors. I might change this and say, see prior, like a broad understanding of prior work that has been done, right? I could say that. I said instead what has been tried because I want to specifically find out what experiments have been done. So I'm gonna try to look for experimental research papers. And from the experimental research papers, I'm gonna see if there are any professors I can contact, okay? So two kind of objectives here. Okay, so before I switch over, things to address when walking through an example paper, or not to address, but I guess to ask. First of all, think about what do papers provide that textbooks and Wikipedia doesn't? Okay. What do you do when you're confused about something in a paper? What are some Google resources you can reach out to? How do you skim the paper? How do you determine what things you look more into? How do you interpret the figures and pictures? So I'm going to try to answer most of these points while I'm looking. Okay. So, okay. Do you see my whole screen?
Yep.
Okay. So I'm on a website called lens.org. I linked it a few days ago. I rediscovered it. I found it a couple of years ago and then I forgot about it and I rediscovered it and it's actually pretty cool. So I have that. This is one website that you can use to find papers. You can also use Google Scholar, right? There's also something called semanticscholar.com or .org. This one is AI powered. So sometimes it finds things that Google Scholar or lens.org can't find, and it's very interesting. And there's a whole bunch of other stuff, like there's also ResearchGate, which is like a social media of papers for scientists, is the best way I can think about it. You can search in any of these. I'm just going to use lens.org, okay? And I'm going to search for my topic, which was microfluidics and synthetic biology, right? I'm going to search microfluidics, and then I'm going to use, okay, I'm going to make it bigger. There we go. Oh. Okay. So I'm putting synthetic biology in quotes. This is like Google, Boolean searches. I don't want synthetic and biology to be separate. I want synthetic biology, that to be the keyword. And then I'm going to put microfluidics. Oops. Microfluidics. Okay. So I got 487 hits. I really like Lens because it has so many filters. I can even filter by institution and literally search up UW if I want. Oh, that's sad. Nothing comes up. Wait. Oh, there you go. Okay. I can do that if I want. This is all from UW, all 4 of these. And that's actually pretty cool. It turns out, so I'm already going to start thinking out loud. So I just did that because I thought, ooh, it would be nice if there's somebody at UW, right? There's only 4, so it's not that hard to look through these. I'm just looking at the titles and I'm thinking there is no mention of microfluidics in any of these. This one is really broad. This one is very specific. So this is probably a research article. And here, when it looks at the match, so lens.org will show you where in the text it matches. It says, on quartz surface in a microfluidic chamber, where a single molecule observation is going to be made. This sounds like a methods thing, where it's probably a sentence from the methods section, and they just happen to use a microfluidic device. It's probably not the main thing they're looking for. So I'm going to skip this. This one, I'm not so sure. This one, I'm not sure either, but I know what a nanopore is. So nanopore is not microfluidic, so I'm going to skip that. This one seems like a very broad term, bridging the gap between systems biology and synthetic biology. I'm going to bet that this is a review, but it's a review on systems biology and synthetic biology. I know that systems biology is about computational studies, not about wet lab work. So I'm going to skip this, but what if I didn't know, right? So maybe I will literally just Google systems biology. Okay. And I'll just read the Wikipedia article for a couple of minutes. And I find out that it's a computational and mathematical analysis, right? So then I'm going to think, no. Okay, so I'm not that confident in these four, so I'm just going to take out this filter. Okay. I can search this. Okay, so now that I took out that UW filter, now the titles look very relevant, right? Microfluidic technologies for synthetic biology. That's great, right? So I'm going to open this. That's exactly what I want. The problem with lens.org, it doesn't link you to the actual article, to the actual paper, unless it's open. Oh, yes, it's open. Great. PDF. Okay. So now what am I gonna do? I'm gonna try to look for an actual specific experimental research article, right? That's what I said. This says review. So, okay, well, that's fine. I might read this later, but I'm not going to read it now. Okay. So I'm going to look for a specific article. So I'm going to just keep on doing this. I might sort by citation, right? Because that means that it's a pretty famous or popular work that people like citing. I might do something like that. I did this a few minutes ago, and I found some relevant articles. There it is. Okay. So I found this one. Inhibitory effect of common microfluidic materials on PCR outcome. So I'm using Sci-Hub because I don't have UW access anymore. And this gets me articles for free. You can wave at her. She's the person who made this website. So this is very specific. Why do I know that? It looks like that like invertebrates article that we were just looking at. It's specifically targeting one very precise thing that it's looking at. It's looking at the inhibitory effect of materials for microfluidics on a process called PCR. Very specific, right? It also follows the regular format. That's good. I think this is a regular article, right? Regular research article. So what am I going to do first? Well, I just looked at the title. Again, that's what I look at first. So it's looking at common microfluidic materials on a process called PCR and seeing how they can make the process worse, inhibitory, right? Okay. I'm looking at the abstract. I'm skimming it. Most of this glazes over for me. So I'm just going to go back to the first sentence and read it. In this study, we established a simple method for evaluating PCR compatibility of various common materials employed when fabricating microfluidic chips. Okay, great. So microfluidics, if you didn't know, is sort of a process. Actually, I found another thing. That looks like this. It uses very small channels to do like micro experiments using fluids. So that's microfluidics. And they need to make these using various different types of materials, right? So This paper is looking at how do these materials affect this process called PCR. Okay. So I don't know PCR. Let's pretend. Well, I'm going to look in the introduction for that. I immediately went to the introduction to see if they have a description, because the introduction introduces various concepts. There it is. Polymerase chain reaction, PCR, is a thermal cycling procedure for amplifying target DNA. So it's a procedure in molecular biology for making multiple copies of DNA. Most of you guys know that. It actually says in the next sentence, actually, it just caught my eye, the PCR compatibility of silicon, silicon dioxide, and other surfaces have been studied. However, the results are inconclusive. So there you go. It's actually already saying in the introduction here what has been done before and what hasn't been done, right? So this suggests why they are doing this study in the first place. Another thing that caught my eye in the introduction is this thing. In most cases, adding bovine serum albumin effectively improve the reaction yield. So I'm curious, and I'm thinking, why does adding that thing increase the reaction yield? Is that even true? So I'm literally going to search this up on Google. I'm not going to do this for too much longer. And there it is. There's actually a company that sells it. Actually, I'm going to search it up with PCR. PCR. There you go. Thermo Fisher is a company that sells scientific material. It actually says here, bovine serum albumin, there we go, is ideal for stabilization of enzymes during storage and for enzymatic reactions where the absence of nucleases is essential. So essentially what it's saying here is that, what it actually says here, BSA increases PCR yields from low purity templates. So that's good. I wanted to check to see if this thing is something that they studied or it's already known. It probably is something that's already known, right? I am going to skip to what did they do. So I'm looking at this figure. I'm literally skipping to just looking at the figure. So if I wasn't in wet lab, I wouldn't really know how to interpret this. So I might ask somebody in wet lab, but just looking at it, they have little tubes. They have something with BSA, something without DNA, something without polymerase. If I don't know what polymerase is, I might just search it up. They have all these things. They put it on ice. So what am I trying to get out of here? If I looked at the figure for a couple seconds and still don't get it, I'm going to move on to the caption. The caption is usually pretty good. The PCR compatibility assay. So that's what they're describing here. Both the total reaction inhibition experiment and the DNA or polymerase absorption experiments are described. I don't really know what that means. The PCR is performed using bench thermal cycler, so that's a machine, and subsequent imaging is done in agarose gel. The control experiment was done running PCR without adding any of the material. Okay. So this still doesn't really tell me what it's trying to find, but I guess this is trying to show the inhibition and it's an experiment to prove that. So I might actually look for a description of this in the text. So what I'm going to do is, this says figure 1, right? So I'm actually going to look to see where in the text it refers to figure 1. And it actually refers it here. So I might actually read this section, this paragraph over here, to try to find out what are they doing here. Okay, let's say I'm done with that. There are some results. Okay, more results. I might want to look at this later. I don't want to look at it right now. I just don't feel like it. I'm going to instead skip to the conclusion. Or sorry. I'm going to go back to the introduction actually and try to find out what exactly were they trying to test and prove. So I'm going to go to the end of the introduction. That's usually a good place to go to. And I'm reading here, to assess the material inhibitory properties, we decided to perform only blah, blah, blah. It seems like they're still talking about very detailed, why did they do certain things, which is not what I want to find. I want to try to find a thesis statement. PCR reaction optimization through choice of surface materials is of the utmost importance as it enables. No, I don't really want to hear this because this is talking about why this is important. I want to hear about what the thesis statement is. Our assessment of blah, blah, blah is also pertinent and beneficial. Okay, so they don't actually talk about the thesis thing here. I'm a little bit bummed. I might try the conclusion instead. So I'm going to go to the conclusion. And actually, the conclusion is a lot better. It says, a problem, however, is a PCR inhibitory effect due to the interaction between PCR reagents and the surrounding environment. So that's the problem that they identified. Some materials are poorly standardized with batch-to-batch variations. In this study, we introduced a simple test for assessing the compatibility of materials in PCR. Because of its speed and simplicity, we have been able to compare the PCR compatibility to a wider range of materials than any other previous study. So actually, this study is not about the materials. It's actually about, we created a better, simple test for testing the materials. So now I figured out, what is this paper actually talking about? Okay. So that's sort of my walkthrough of how to look at papers. Okay. So you can see that I'm skipping through multiple different parts of the paper. I'm thinking through many different things that I'm looking at. I'm reading some things, I'm skimming some things, I'm reading some things in detail. That's sort of how your literature review should look like, right? And as you're doing this, I totally recommend you guys to go into your research details document for your topic, right? So I'm just gonna show you right now. So these research notes, this is where you should keep all your notes from what you've gained out of certain paper, right? So let's pretend that, I don't know, microfluidics was something that we're doing, then you would write the takeaways from this paper in that document, right? And maybe your thoughts about pursuing something similar to this, or why was this helpful, right? By doing that process that I just kind of walked through, you might at some point realize this is not what I want to be looking at. At that point, just stop looking, right? One last thing, I also wanted to see people that I can contact, right? So let's assume that this is something that I really want to look into. This is really relevant. Well, the first obvious place is, who are the researchers, right, that did this paper? So there's a whole list where they're from. They even have contact information here. Great, they've done all the work for us. We could contact them if we want to. Let's say we want to contact somebody more closer to us. They're from Saudi Arabia and Hong Kong. I'm going to look at the references section, or I might actually look at, so this is actually really nice. This paper actually has biographies. So I might actually look at the biography and spend a little bit of time seeing if anybody has done research in the United States, actually. This person did stuff in Canada. I don't know if PhD in electrical engineering is that related to the field, so maybe not. Okay, maybe this isn't going to work. I might actually look at the references instead, right? And by looking at references, it's not choose a random thing and click on it. It's, I'm going to go back to the introduction and try to see which citations are used for certain key points that I want to know about. Let's say, I don't know, for some reason, I know what FTIR is, and that's relevant to what I'm looking at. So I'm going to go to citation number 7, and I might look into this. I might literally find this paper, right? It also said 13 to 16. So I might actually look at these papers too next. And maybe some of these people might actually be better contacts. So that by doing that, you can get tens and tens and dozens of contacts for people that might be experts in this specific thing, because this is what I want to look into. Okay. So yeah. Okay, so it's 7:55. That was a lot. I just kept on talking. Do you guys have any questions?
I had another question.
Yeah.
So in the abstract, especially for this paper, it made it
Mhm.
seem like they were gonna talk about the different materials, but then in the conclusion, like you mentioned, like they were sort of talking about their own technique. Does this mean like we probably have to read more about the paper to know what they focus in more on, right?
Right. So it could be that I was seeing if they do talk about the actual materials themselves, but it doesn't seem like it. So um, I don't know. This might mean various things. It might mean that we need to look more into it, or this um, abstract, when they talk about the parts, I actually was reading just about the introduction. And they basically wrote a lot of introduction in the abstract instead of talking about what they actually did.
Mhm.
That's my guess, actually.
Okay.
Um, so actually, now that we've analyzed the paper a little bit, this is where they actually start talking about what's relevant for the paper. Do you see
Oh,
that?
right. Okay.
Yeah.
Yeah, I see it.
So they didn't do a very good job of writing this abstract. Again, quality differs. But yeah. Any other questions?
Uh, so when we're taking notes on like takeaways, should that be done sort of like after we read the abstract and then look through the results and discussion, or should that be done like along the way?
I think it should be done along the way as you're reading a paper. So some people just some practical tips. Some people really like to print out the papers and actually like highlight physically. You can also do this by using. You can also do it digitally by using various programs. I don't think I have it here actually, but there's a program called Zotero.
Oh, right. Yeah.
Um, and what it does is it organizes all your PDFs for you of papers, and you can open it in Zotero, and there's like a highlighting section, and you can like search up all your highlights and stuff like that. It's really really useful. I recommend it. Unfortunately, it doesn't work with Teams. It's solo, but yeah, I suggest that. I would just do whatever makes sense for you. If you want to read a whole paper and then write your thoughts, or write some thoughts while you're reviewing it, that's fine too. Yeah.
Okay.
Okay. So for everybody else except for Risha, was it helpful?
Yeah, it was very helpful. Thank you so much for doing
Okay.
it.
Okay, that's good. That's good.
Yes, that was very helpful because now I know like which part of the paper to look more into
Mm-hmm.
and when reading. So thank
Yeah.
you.
Yeah, that was definitely useful. Now I kind of have at least a direction to go in when I'm approaching a paper. So
Okay. Okay, that's good. Yeah, and so I have a final slide here that I just want to end on. I know we have one minute left. So these are just some tips and points. Try lots of search terms if it's not coming out. Trial and error is science, so it might be frustrating sometimes when you're doing literature review. Sometimes there's dead ends where you just can't find anything. Um, Google is your friend. Don't be afraid. And definitely, you will feel like you're dumb. I just want to point that out, that you will feel like you're dumb when you're reviewing research, but appreciates the fact and be humbled by the fact that you're looking into amazing, cutting edge, bleeding edge research. So don't be disillusioned. It's definitely a process that the more you do, the more you become more comfortable with it. So yeah. And also, if you ever have just if you're not sure if you're doing something right, or if you don't know what's going on, or if you don't understand a certain paper, that's what Zulip is for, right? So ask on Zulip if you have a question about a certain paper, if you don't understand it, if you're like, does this actually mean that? I am totally, totally happy to actually read it as well and try to give my own opinion, and Carl as well. That's also part of project development. Like you can ask about that as well. It does not have to be a solo activity at all. Okay, cool. Any last minute questions? Okay, great. I'm gonna stop.